Chemical Societal. Oh, okay. Anyway, this is the California section of the Women Chemists Committee. And we're having our quarterly meeting today. And first, I want to acknowledge the many volunteers who work to make this meeting possible. One is Michael Chang, and he is, you can see him. He's responsible for the contest that we're going to have. I'll tell you a little bit about that later. Catherine Louie comes all the way from Texas to help our Women Chemists Committee.
Catherine.
She's responsible for the recording of it. And then Laurel Wood is a new volunteer. Laurel comes from a university, okay? So she's young, not like us, okay? She's young. She's volunteering already.
Don't say that.
And then we have Fanny Frausto, who she can't be with us today, but she does the YouTube production. And then we have volunteers who also can't be with us today, but they play a very important role. Tony Miao, Mina is here today, and she has put together programs for the Women Chemists meeting, and then Sheila Ye, okay?
Sheila, yeah.
Yeah, and Sheila is responsible for putting together the flyer that you all see in various parts of the country, and that entice you to come to these meetings. Without her, this wouldn't work. Because I don't know, remember, I don't know how to do anything except find speakers, okay? That's the only thing I know.
That's awesome. That's pretty important.
Totally useless. Okay. Now, speaking of speakers, let me tell you a little bit about the speaker. The speaker that we have today, Marya Lieberman, anyway, she responded to my email request to please come and talk to us. And how did I find her? You know that C&E news that you get every week? That's how I find her. I found her because I didn't know she existed. Okay. Now, since then, I've learned that you'll be really educated today. This is a professor told me this, okay? One of your pals, actually. She's not here today. And then I also learned a little bit about her. You know, Maria got her first taste of chemistry when she was in her stroller. She was probably exposed to a lacrimator. She didn't know it was a lacrimator, but she was probably exposed to it during this free speech movement. So here she is in her little stroller doing chemistry, right? At that age. Then after that, well, she got interested in chemistry and did her undergraduate work at MIT. And then she went to graduate school at the University of Washington. Now, what's really interesting, after she got her PhD, she went to Caltech to do her postdoc. And Caltech was absolutely flummoxed. They did not know what to do with Maria because she was pregnant. Okay? So they didn't know what to do with her. And she probably has great stories about that. But nevertheless, she finished her NSF postdoctoral stay at Caltech, and then she went to teach at the University of Notre Dame, and she's been there ever since. And by the way, she went there with her husband, so somehow she picked up a husband between the time she was in undergraduate to graduate school. So she picks up this husband, and they have offices next door to each other. Okay, now isn't that something? All right, so then she's had a very varied research career, but what she's going to tell you about today has to do with paper microfluidic technology applied to fake drugs. Now, as you can see, I'm old. I'm really interested in this subject because I take drugs. And when I read in C&E News that there might be fake ones, I mean, I kind of got nervous because I mean, I'm taking drugs. Okay. I don't want mine to be fake. And I'll bet you no one else wants them to be fake either. Okay. So without further ado, I'm going to let Marya start her talk.
All right. So let me see if you're going to have to give me permission to share my screen.
Oh, yes.
Here we go.
Where do I do that?
Good. But thank you for that very nice introduction.
I gotta get to more. No.
Share. Yeah, it's under share.
It's under share. Where is share?
Little green arrow thing.
Oh, here it is. I'm sorry, I didn't see it. Share.
Okay, now you got to give me permission to share.
Oh, wait a minute. Somebody. Yeah, you don't want. So I don't want to share, but you have to share.
Right.
So I'll stop sharing. How do I get you to share?
Click on the name and then the left or right click and there's allow to share or something.
Okay. The name. Participants. Yeah. Okay. Thank you, Michael.
I love that this is being solved collaboratively.
It has to be because I'm such an idiot. I mean, you know, where are you? Here you are.
There's 3 dots. Click the 3 dots.
I did click it. So do I make her a
co-host?
Yeah. Okay. Thank you, Michael.
Okay.
I think it's going to happen. Sorry, Marya. I told you the only thing I know how to do is to find people.
Well, we've managed.
Then can you also make me a co-host so I can?
Oh, yes, Michael, I have to make you a co-host because, okay, I forgot to talk about the contest. You see, in our section, we hold a contest after the speaker speaks based on the subject that the speaker has talked about. And so you'll get 3 multiple choice questions from Michael, okay? And if you want to participate, it's all volunteer. What you get to do is you have to answer the 3 questions. Anybody who answers the 3 questions correctly, all multiple choice, will get a prize, okay? And it's an ACS theme prize. And when you answer your questions, please send your answers only to Michael and use the chat box, okay? You don't want to send it to everybody because if you do, that would be not the point, okay? You are trying to actually win a prize for yourself, not for everybody else, but for yourself. Okay, sorry, Marya.
That's great. All right, can everybody hear me? Is that working?
Yes.
Audio is working. Okay, so I like talking on Zoom with some interaction going on. And so what I'm going to suggest is that people use the chat to put comments and questions in. And there'll be a couple of natural breaks and maybe we can do, we can talk then if there are questions or comments. Okay, so that I don't just lecture, because I'm a university professor, so I can talk for 50 minutes about anything. So here we go. All right, so I want to tell you today about how I'm using paper microfluidics to find substandard and falsified medicines in low resource settings. And So let's, I hope I'm going to get through a couple of demonstrations. You're going to see a lab tour. It'll take you into my lab. And this problem that I work on in developing countries also impacts the US, mostly in the form of the opioid crisis that's affecting every city and town in the US. So here we go. All right. So in 2017, the World Health Organization did a big review of the evidence on the quality of medicines, and they're focusing on medicines in low and middle income countries, which the jargon for that is LMICs. So if you hear me talk about LMICs, that's what I'm talking about. They found over 100 published studies that involved more than 48,000 samples of medicines that were collected in LMICs and then analyzed in different ways. And out of those samples, about 10 to 12% of those samples failed some kind of quality standard, most often the HPLC analysis or assay. What that means is that 1 in 10 medicines that's being marketed globally is of poor quality. And that means that so people are spending on the order of $300 billion US a year buying those medicines. And so if 10% of them don't meet standards, then that means $30 billion is going to companies that probably shouldn't be making that money, right? If they're selling low quality medicines. And the WHO has also done some economic and health outcomes modeling. And those preliminary studies have focused on just 2 problems. One is malaria, and the other is community-acquired pneumonia. And those diseases in developing countries are some of the main killers of children under age 5. And so the contribution of low-quality medicines to the death toll is believed to be nearly 200,000 a year. So those are a monetary cost, but also a cost in human lives. Well, I started working on this problem in 2010. I worked with some faculty at Notre Dame who were involved in international research, and they were getting involved with this AMPATH Research Center in Eldoret, Kenya. And they brought me there as kind of a sample faculty member. So I got to go to Kenya, which I'd never been to Africa before. But I really enjoyed going there, meeting people, and that has developed into research collaborations that I'm still working on today. So here I am at the AMPATH Research Center in Kenya, and the folks in this picture are myself, at the time, an undergraduate researcher, Sarah Bleasy, who was doing an REU with me, and she later became a grad student with me. And then 2 of my collaborators in Kenya are Mercy Mena, who's a pharmacist, and Felix Were, who's a pharmacy tech. And also he's in the business end. So I've been working with them for more than 12 years. And I want to start out by just doing a quick definition. So I've said that I'm interested in finding substandard and falsified medicines. And I just want to give a quick overview of what that means. So a substandard medicine is a medicine that doesn't meet 1 or more of its quality standards. So every medicine, if I want to sell a medicine in a country, then there's going to be this whole laundry list of quality characteristics. It's called the pharmaceutical monograph. And that pharmaceutical has to pass all those tests to be of adequate quality. And so if a product fails any of those, it's substandard. The most common substandard products that we detect are products that have the incorrect amount of the active pharmaceutical ingredient, or API, in them. So they might have too little, they might have too much, but in either case, it's not going to give the optimal therapeutic profile for the patient. So a falsified product is a product that is misrepresenting what it is or who made it. So that can include a product that contains the right API in the right quantity, but the name of the manufacturer is not correct. So that would be like a counterfeit Viagra product, for example. It might have the right active ingredient and work just the way it's supposed to, but it might not. Okay, and then the other kind of falsified medication is one that says on the package, I contain 100 milligrams of doxycycline, and then you go and you test it, and it actually contains some other drug. Maybe there's no doxycycline at all in that product. Okay, and there's some
sorry,
overlap.
we're still on the screen that says AMPAS Center. Are you
Yes.
in other slides, or is this still the same slide?
Yes, that's correct. I've got another slide coming up though, which has an example. So
I
here
see.
comes. Here's an example. So this is a product that is a substandard product because it doesn't contain the right amount of the API, and it's also a falsified product because it contains an excipient that's not listed on the package. So this is an amoxicillin product. So we found this is a Kenyan product called Caremox. And this product is available in 2 different dosages. One of the dosages is a 500 milligram capsule, and the other is a 250 milligram capsule. And you can see these are 2 volumetric flasks that have been made up with 50 mgs of each of these product, because we were preparing to do HPLC analysis. And you can see, so on the right, we've got a normal amoxicillin product. Everything is soluble in water. And on the left, you can see the water's really cloudy. And that's because the manufacturer of the product on the left thriftily put about 50% of the amoxicillin is replaced with talcum powder. So that's a substandard and falsified product. Okay, let's take a look at the health impact of that drug. So this product was very widely distributed in Western Kenya. We collected it in multiple counties of Kenya, and it was about 43% of the market for amoxicillin products. So in the US, we use amoxicillin for treating like kids with earaches, and we see it as being not a very important drug. But in Kenya, this is the main treatment for community-acquired pneumonia. And community-acquired pneumonia is like the #3 killer of children in Kenya. So for severe pneumonia, the death rate is about 2%, even if the kid gets treatment. And if their dose of amoxicillin is at a 50% level, which it was in this product, then that survival rate or the death rate doubles. And so we had a rough idea of the market prevalence and the number of kids under age 5 who get pneumonia in this part of Kenya. So we estimated the impact on the population, more than 1,000 excess deaths of children under age 5 from this product. Now, it's something kind of interesting when you look at the fine print on this product. So Caremox is a Kenyan company. They're located in Nairobi, but they do not make this product. When you magnify the fine print on the package, what you can see is that the capsules were actually manufactured by contract pharmaceutical companies that happen to be in China. The 250 milligram dose was manufactured by one company, Shaanxi, and the 500 milligram ones was manufactured by Shandong, Shenglu. And so it's interesting that one of these products was substandard and the other one was fine. Okay, so what this means is that when you're tracking these bad quality products, you need to be tracking the manufacturers. of the products, not just the brand names or the distributors. You have to track down to the level of the manufacturer. This is another product that we found in an East Asian country that I can't talk about because the regulator in that country is very sensitive to discussions of their country being involved in bad quality products. We believe that this is what's called a deceptive counterfeit. So this is a product that is imitating a genuine product, but what's inside these capsules, there's no amoxicillin at all. It's just chalk. Yeah. So without treatment for severe pneumonia, the death rate is about 6%. In this Southeast Asian country, in the region where this medicine was circulating, we figured as many as 150,000 children under age 5 may have been exposed to this drug that would have caused about 9,000 excess deaths. And this country happens to have some huge refugee camps and I think that that's probably why we found this counterfeit product in this part of the country, is that it may have been going into that location. Okay, so now I want to get some comments from you guys in the chat. So if you're, I hope everyone can see at the top of the screen, you can see a little chat bubble on your Zoom. So if you just click on that, that'll open the chat panel. And what I'd like you to do is just look at these two lab settings. One is a commercial HPLC lab in the US, and the other is a lab in a salt factory in Haiti. One of my, Marco Magellan was an undergrad researcher with me. He went and visited this lab in 2011 when it was safer to be in Haiti. And he was helping the salt factory to analyze their salt iodization. So just in the chat, just think about some of the differences that you see between these two labs and drop them in there. And I'll have to tell you, there's one difference that the undergrads always notice first. And we'll take a couple minutes so people can put things in. Yeah. Yep, great comments. Yeah, Lori put in a comment about the lighting, and the lighting is, there's no electricity in this lab in Haiti, so they're doing their titration by the window. The person who's doing the titration is actually not 6 years old. He's a high school teacher in Haiti, so he's just kind of crouched down so he can read the level in the burette. Great. Okay, so the thing that the undergrads always notice is the Smirnoff bottle. That's the lab alcohol, because in a developing country, right, you're in a low resource setting, and one of the resources that's hard to get is supplies, right? So solvents and reagents. The lack of PPE is, I see several of you have noticed that. The difference in the equipment, well-equipped US lab versus the sort of very basic level of equipment. Yeah, and of course, if you're doing HPLC, then behind the HPLC, there's a whole lot of support for the instrument. There's people who can come and fix your HPLC if it's broken. There's a way to get new columns, right? You can just order a new column and it arrives in 2 to 3 days in the US. That is not the case in a low resource setting. Okay, so became interested in, so I had these students who were working on the salt factory in Haiti, helping them with their, to produce their product. And I became interested in how can you analyze pharmaceuticals in this kind of situation? And so the system that I wound up designing and developing uses paper microfluidics. So let me show you sort of, I'm going to show you, and then I'm going to go in a lab and do a demo. I can see if I can change this. Okay. So this is the device that we wound up developing. So this is called a paper analytical device, or PAD. It's a piece of heavy chromatography paper, and there are dried reagents that are stored on the pad. And so that means that the person who's using the pad, all they need is the pad. They don't need to acquire reagents and special solvents. The sample is applied as a powder, and so that means you can test dosage forms like capsules and tablets. You don't need to make any solutions, so you don't need any volumetric glassware. You don't even need a balance to do this test. As long as there's some powder in each lane, then the chemical reagents will do 12 different color tests on whatever's been applied to the card. And so we do the test by putting the base of the card in water for a few minutes. And then the water runs up the lane, and you can see that this region, which is the color barcode region, develops some interesting colors. And so this particular barcode here has some chemistry going on. The drug that that is in this picture is ciprofloxacin. So ciprofloxacin has tertiary and secondary amines, and those react with cobalt thiocyanate. So this is a version of the Scott test, which is used in police labs to detect cocaine. So it's a general tertiary amine test. And then also there's this 1,3-dicarbonyl group, and that can react with iron, and it forms, basically it's like an iron tris-acac type compound. And for that, we get some variation in color intensity depending on the amount of ciprofloxacin that's present. So we have some ability to quantify this way. So we call that the color barcode for this drug. Okay, so I think I'm going to take you into lab in a minute. So let me just show you, this is what happens with ciprofloxacin. Well, what if we ran a different type of drug?
Can I ask a quick question?
Yes.
So this is kind of like the fingerprint region of the IR. Is this this is what the positive test for this drug would be, and so they use that as a reference?
Yes.
Gotcha.
Yes, that's right. And actually I'm going to get into a little bit when we're doing a lot of computer aided image analysis. But for a person who knows what types of what these color tests do, you can often just read the card just by eye, just by knowing, what different color tests do. So let me show you another example. So this is ciprofloxacin up at the top, and the barcode for ciprofloxacin, that test for the tertiary amine, and then in lane L is the iron complex with the 1,3-dicarbonyl. Well, the bottom drug is isoniazid. So isoniazid contains this acyl hydrazine, and so this terminal amine, the reactivity is very souped up. It's like a supernucleophile. And so that activates several new lanes. So lanes G and H are lanes that respond to strong nucleophiles. And then lane B is a ninhydrin test. And normally ninhydrin will give you a Röhrmann's purple, which is a blue color. But we found that if you do the ninhydrin test at room temperature, you get stuck at an early intermediate, and you form Schiff base compounds, which can range in color from yellow to orange to red to green. So lots of different colors instead of just Röhrmann's purple. So so this is one of the interesting things about these color tests is that we're not just getting a yes, no response, right? We're getting some more subtle or nuanced information, which is really useful. Okay, so I think it's time for a lab tour. You guys up for that?
Mhm.
Okay.
Yeah.
Mhm. Mhm.
All right, I'm back. Sorry, I unplugged my computer and all hell broke loose, but now we're back. Okay. Here we go. All right. So my lab, and this room in here, this is where we keep our pharmaceutical samples. So these are about 3,000 samples in there right now. So these are samples mostly from sub-Saharan Africa. So I'll take you in, show you. So down here, these are all chemotherapy drugs. Most of these upstairs are antibiotics. Sorry, it's pretty noisy in there because of the refrigerator. But here we'll come. Okay. So we're in lab and I wanted to do a test with you guys. So first I want to show you how we make the paper test cards, and then we'll run one and you can see how it works. So paper test cards start with just a piece of paper. So this is a heavy grade of chromatography paper. We feed it through a laser printer and we print 8 of these cards are printed on the paper. And so this is just a QR code. Each of the cards has its own serial number. So you can see we've made a lot of these cards. We're up in the 79,000 range right now. After the card is printed with the laser, it next goes through a wax printer. And the wax printer lays down ink that is like, the ink is like a big crayon. It fits into the printer. It's a Xerox color cube printer. And we print it on the front and we print it on the back because this chromatography paper is a 400 micron thickness. Deborah, did you have a question there? I thought you were raising your hand. No. Okay. So the wax ink goes on the page and then we stick it in an oven and bake it for about 8 minutes. And you can see that the ink has kind of smeared out. So that the ink soaks into the paper. It's kind of like a kindergarten art project. Okay. So, but what that does is it creates a hydrophobic barrier on the paper. So now we can build our paper microfluidics with wax and with cellulose. And so you can see here, this has had the reagents applied. The way we put the reagents on is we just get this is a multi-spoke inoculating manifold. So this is used if you have a 96 well plate, like one of these jobbies. If you're growing a bacterial culture and you want to replicate it, then you can just put this in here and put it in another 96 well plate and replicate your cultures. But we found if we put the reagents in here, we can just stamp them onto the paper. Okay, and that puts those the reagents on pretty well. So you can see that in each lane there are different colored areas. Those are the different reagents, and the wax ink does a pretty good job keeping each one in its own lane. So we can just stamp these out, which is good because we are using thousands of them. Okay, let me see if I can, I think maybe Elaine, you may have to make me a host again. I think I probably fell off the hosting. And let me just see if I lost my presentation. I think my presentation needs to be restarted. Let me see if I can do that. Okay, here we are. Thank you, Elaine. I think I'll be able to share the screen. Let's see, how's that working? Okay, I think that's sharing now. Good. Good. Yes. And Lori, yeah, this technology, I didn't invent this technology. This is Whiteside's wax printing technology. So he published those papers in, I think it was 2009. But actually, it turns out there was a patent in 1943. I think there was a patent by a Midwestern chemist who dipped metal circles in paraffin wax and stamped them onto paper because he wanted to do spot tests more accurately, with a nice defined region. So I thought that was pretty cute. So, the task that we're often dealing with in a field situation is we have a product and we're trying to figure out if it's the correct API. If it's not, then it would be a falsified version. We want to also detect whether maybe it has some of the correct API, but it's cut with a filler. So that would make it a substandard or falsified version. And then, of course, the last possibility is maybe there's no API at all. So in order to do that, we're going to use the test card. So we're going to take this card here and load some powder on. And I'm going to do ciprofloxacin with this card. We can do about 20 different pharmaceuticals with the current version of the card and the app. So, of course, there's an app. So let me show you how to do this. So first I will load it. Let's see. Okay. So I'm just using this highly sophisticated material here. This is a little bamboo coffee stirrer. Take out the sample and then just smear it across like so. And just tapping off any excess, and then you can see that there's some powder on each lane. So I've got powder on each lane. Yay. So now I'm going to dip it in water and let it develop. So here's my little water dish. Just stick that in there. And okay, so it's going to take about 3 minutes to run. So while it's running, I'll talk some more. Okay. So that's running. Okay, and then I'm going to give you guys a preview. This is what we're looking for. So for the result, remember we have that barcode. We have the tertiary amine signal, which is going to show up in lane D. And we have the signal from the dicarbonyl should show up in lane L. And if we have some bad stuff in there, say someone has used starch as a cutting agent, then we'd see starch showing up in lane J. So that is the starch iodine test producing that black color in lane J. So we found that iodine, of course, is not, you have to make triiodide to get it into aqueous solution. And triiodide doesn't last very long on the paper because there's an equilibrium and the iodine can evaporate. But if you mix it with a polymer, povidone, the povidone sort of encapsulates the triiodide and it's stable for more than 2 years on the paper. And the iodine is still reactive with the starch, gives very nice signals if you have starch there. So if you have ciprofloxacin and starch in your pill, then you get a sort of a superposition of the signal from the ciprofloxacin, where you get the blue color in lane D and the orange color in lane L, and you also get a starch signal in lane J. So to some extent, these colors are additive, and that's very handy. So, all right, so this card hasn't developed completely. We're going to take a look at it now, though, because you can see some of the colors are already developing. Okay, so for example, you can see in lane D, you can see that nice blue color showing up. That's the tertiary amine color. And then in lane L, the iron tris acac is developing that orange color. Okay. And in this sample here, there's actually no starch. So lane J is a brown color rather than a gray or black color. And that will.
I can't see the video. I don't know if I'm just missing a window.
Oh,
I'm seeing your screen.
there's the video is just showing up in my tiny talking head panel.
Oh yeah, I don't have that shown. Okay, thank you. I'll go find that.
If you can show, I'll hold it up so you can see that. Oops. Okay. How's that?
I found it. Thank you.
Okay. Yeah. Too many panels. Okay, so I'm going to put this back in the water and let it run a little more. I want it to be fully developed because in a minute I'm going to show you the phone app and we'll try and read that. Hopefully it'll at least figure out that it's ciprofloxacin. So one of my senior colleagues when I started out in this job told me that whenever you do like a class demonstration, you have the chance to make a big fool of yourself. So I always feel like I always remember that when I do these demos in a talk, but we'll see. Okay, so far this is a nice little academic project. We developed this device. We did sort of a lab validation, and we wrote a paper and we published a paper about it. And but that is not where this project stops for me, because I don't want to just publish papers about this. I want to have a real impact on the substandard and falsified pharmaceuticals that are circulating in all these countries. And so what this has done is it's really pushed my work more in a sort of a chemical engineering department direction. So yeah, sometimes I feel like I'm not really a chemist anymore. I'm really worrying about these de-risking things, you know, is this device manufacturable? Is it easy to use? Do we have IP protection? Usually these are things chemists are not thinking about. We've been working a lot on building partnerships with regulatory agencies in developing countries. That's not always easy. Those folks have a lot of responsibility and not a lot of resources. And so when Westerners come in and say, oh, we're going to save you, they often are not really thrilled about that. And you really have to show your bona fides through your work. All right, let's take a look. We are doing field testing. We are doing neural net training. Both of these things take thousands and thousands of pads. And so we've worked a lot on the manufacturing. So we currently have an alternative manufacturing method we're using. So this is a prototype. This is an air gap pad. It has only 7 lanes instead of 12 lanes. But on the other hand, in 8 hours, they made about the equivalent of 32,000 pads with this technique at my collaborators at Serum. So yeah, so that really drove home to me the difference between making devices one by one in a lab and mechanizing the manufacture. So I've been, so we're working on taking that through to the reagent deposition. We've been doing large scale field tests with drug inspectors. So this is sort of our target market, is people who already are tasked with doing surveillance of their markets to detect substandard and falsified drugs. So my now graduate student, Sarah Bleasy, and I went to Tanzania in 2014, 2015. We worked with 20 drug inspectors, trained them how to analyze, test card the drugs with the pad. And then we sent them 3,000 samples and 3,000 pads and had them analyze the former with the latter. And they were able to classify the products as real or fake with 93% accuracy, which is pretty good. My grad student, Sarah, was at 97% accuracy. So she was more accurate than the drug inspectors. But this also got us 3,000 pad images that were taken with 20 different cell phones. And we took that and took that to one of my colleagues who is a computer scientist to see if we could use machine vision. So he trained a neural net to read these images, and the accuracy of that was also 97%. So we said, aha, maybe this would be a more usable and more accurate method. So we've started doing this image analysis. So we use both neural network methods, but we also use linear analysis methods like principal component analysis. And so here, this is showing the classification of different types of drugs. So the orange ones that you see that are up at the top of the graph, those are all samples that contain some amoxicillin. Some of them have just 20% amoxicillin, and some of them are 100% amoxicillin. But the principal component analysis is pretty good at differentiating those samples from all of these other drugs that were included in our image set here. And in some cases, like with the ciprofloxacin, the analysis is actually semi-quantitative. So here again, the orange colors that you see, sort of the bars that are sort of towards the top of this little graph, show one of them at the top is good quality Cipro, and then the others are Cipro at lower concentrations. And so this simple PCA approach is not bad at classifying the concentrations. And again, all the things that are not ciprofloxacin are very clearly differentiated. Okay. And then now we have image analysis methods. Now we can take those and put them in an easy to use cell phone app. So I have that app. It's actually out in the world. It's available for both iOS systems and Android. So I'm now going to try it, on the Cipro sample that we just ran. So here's that Cipro sample. You can see the barcode. You humans have already learned to read that barcode, right? Just in a few glances. So here now what I'm doing is I'm setting up the analysis. So I'm telling it what to expect. That doesn't actually affect the analysis, but it will affect how the system classifies. So it acquired the fiducials here. So I'll tell it to accept that. And now it's going to classify it here. So it has done a good job. That's the card. And it thinks it's ciprofloxacin at 100% concentration, which is what the sample was. So, yay. Okay. So, so that's how that app works. Okay, so we're coming up on 3. So Elaine, do you want to stop at this point, or do you want, I can go on and talk about chemo drugs and opioids?
Well, I have a personal interest in methotrexate. Do you have any data on methotrexate?
That's actually methotrexate is one of the drugs that we're analyzing in sub-Saharan Africa. So yeah, I'll go on and talk
Yeah,
about that.
I really
Yeah.
want to know about methotrexate.
Yeah. I don't have any of the analytical data for our methotrexate samples. I think all the samples we collected in Ethiopia of methotrexate were pretty good. The
Oh.
quality problems we're seeing mostly are with cyclophosphamide and cisplatin.
Oh,
But
okay.
yeah, but I can I'll go on. I see that there's some questions in the chat, so maybe I can stop. Oh, okay. So here, okay, so one question from Anne is, do you run a standard at the same time? No, there's no standard. All the references are embodied in the neural net weighting factors.
Hmm.
So if we have a drug that hasn't been analyzed, like a new drug, then we have to collect about 200 pad cards worth of data in order to add it to the neural net. So that's a big pain. So we are gearing up to do it for the chemo drugs though. Lori had a question, how did the card images get transmitted back to me? Are they uploaded when the QR code is scanned? So the images are stored and analyzed locally on the phone. So when you set up the app on the phone, you download the neural net weightings and the PCA matrix that lets you do the analysis. And then that's locally on the phone. But then as soon as the phone has a good Wi-Fi connection, then it transmits what it found to a central database. So this is one of the things we've been really working on is a lot of the pharmaceutical analysis people do in field settings, the analysis results are just stored in paper notebooks. And so it's really hard to connect that information up internationally. And so actually all of this data right now is coming to a central database, and we're connecting that with a sample provenance, like a tracking system that keeps track of all the information about the samples, like where were they purchased, what type of store were they purchased from, was the sample provided in a box, or is it just in blister packaging? So like this is kind of a typical way that we'll get samples. It's like a blister package. This is some of that Caremark stuff. by the way. So this is one of the substandard products. So, but it's in a nice looking blister package, but there's no external box for this. And sometimes we'll get stuff that's just dispensed in like paper envelopes. So what triggers a test from Michael Cheng? That can be like different things can trigger a test. You're talking about when do people want to run a test on a particular product?
Correct.
Yeah, so that depends from site to site. Like one of the ways we use these is I'll have collaborators who are going out and doing sort of covert shopping. So they'll pretend to be a patient. Like in the Kenyan site, we train patients from the hospital. So they do a lot of HIV treatment at the hospital. And one of the problems is that the patients often find it hard to get a job because of the stigma associated with HIV or because they're physically weak. And so some of those people, we train them as secret shoppers, and then they just can go around town. They go to different pharmacies, and then they have a script that they learn about, whatever their medical problem is. And they buy these medicines for us. Question from Mina is, how does the quantifying method work based on color? So Mina, it's kind of like a UV vis spectrophotometry method, right? So it's reflectance rather than transmittance. And often the calibrations are really not very good, but yeah, but that's how it's done. Okay, so I'm gonna carry on. I'll talk a little bit about the work we've done mostly in Ethiopia at this point with chemo drugs. Okay. Oops. Okay, so we are now in the second year of a NIH funded project where we're looking at the quality of chemotherapy drugs in Africa. And so we started last year, and then almost immediately we started finding stuff like this middle panel here. So this is a ifosfamide sample. So it is supposed to be water white, and I think you can see that that's not the color. So that's an example of what we would call packaging analysis or visual inspection of the product. And right now, that's the only method that is available, I would say, basically in all of sub-Saharan Africa for detecting substandard or falsified chemo products. There's no regulatory authority that is currently able to do HPLC analysis of these products. And that is related to the toxicity of the drugs. So we started working in January of 2023, and almost immediately we found there's this fake Herceptin. We had reports from Peru and Brazil of this substandard asparaginase. So this is actually a pretty amazing story. So this is from a paper that was published in 2018. They happened to be doing a study where they were treating children with asparaginase and studying the amount of asparaginase in their blood. And so during this study, they switched from Aginasa, which is one brand, to Luginase, which is another brand. And what they saw was in every patient, the amount of asparaginase activity in the child's blood was below the level that's required for therapeutic efficacy. Okay, so switching from one brand, the blue brand of asparaginase, to the red brand of asparaginase meant that those children were not being, basically their leukemia was not being treated. So these are part of the reason that the cancer survival rates in the US, cancer survival rate, 5-year cancer survival rate for women with breast cancer is greater than 90%. In sub-Saharan Africa, in Ethiopia right now, it's less than 30%. Okay, and part of the reason we believe is the medicine quality issues. The problem is that the chemotherapy products are really, really toxic, and that it makes them very hard to handle in the lab setting. So what we have done is we've developed a special test card. So we call it the chemo pad. The chemo pad is just like a regular pad, except that there's a little plastic cover, and the plastic cover has a strip of absorbent paper. So when the, let me see if I can put this back in the, see if that works. Are you seeing that a little bigger now? Is that working? Okay. So the chemo nurses or the chemo oncologists will prepare a patient dose, and then there's usually a few drops of the drug that are left in the vial, and they can just put those drops on this absorbent paper, fold it over, and apply it to the pad, and then run the pad in the usual way. And so I had my student, this is Madeline Smith, who is at Addis Ababa University at this very beautiful gate at the main campus. She is there doing what we thought was going to be a usability study, just to see if it was possible to use this device in the Ethiopian setting. So, and then pretty much the first thing that happened was this. So she was testing a cisplatin standard, and the cisplatin is tested with a separate chemical test. Cisplatin is a platinum drug, and it reacts with stannous chloride. So it's tin 2 that reduces the platinum, and you make these platinum tin like nanoclusters. Okay, and then those little nanoclusters have a yellow or orange color, and which is very cool. And then, but she was testing this cisplatin product, and the yellow color was very weak or absent. So she brought some of that sample home. And when we analyzed it on HPLC, this is on the left, you can see a cisplatin standard peak, nice peak shape. And the cisplatin dosage form, you can see that it's weaker intensity. And then there's also this sizable impurity peak. That's a hydrolysis product of the cisplatin. We also assayed the absolute platinum content to see if the problem was degradation, and because degradation does not destroy platinum, the platinum would still be in the solution. And what we found was only about 60% of the amount of platinum that should be in that vial was in the vial. So to me, what that indicates is that the manufacturer did not put the right amount of platinum in the first place. So we were able to publish this research. This is a case where I actually delayed publication for a while because Haran Gerba, who's author number 3, asked me to wait on that. Haran at the time was the director of the HPLC lab for the Ethiopian FDA. So I waited for her to contact the manufacturer. I don't know what kind of discussions went on, but I will tell you that in the last 2 years, we have not found this particular product in the public supply system in Ethiopia. So you can make what you like of that. All right, so that's a case where our project impact here is sort of, we're trying to do proactive surveillance of chemotherapy drug quality rather than waiting for people to be harmed by bad quality drugs. All right, so Let me move on. So, this is what unregulated drug supply looks like in the developing world. What does an unregulated drug supply look like in the US? Well, the FDA in the US is, I would say, a very effective and efficient regulatory system. Whenever people complain about burdensome regulations, I'm thinking about my cold room and all the bad quality pharmaceuticals that are in there. But in the US, we explicitly do not regulate street drugs. And so street drugs for us, I think that's our unregulated market. And so the problem with having an unregulated drug market is that there's all kinds of stuff that shows up in that market. And we can't help people who are addicted to street drugs or measure success if we don't know what their problem is. Okay, and I'm going to give you an example in a minute of why not knowing the contents of street drugs can cause huge problems for all kinds of people. So if you know what's in street drugs, then that can help people make better decisions about what they're putting in their bodies and how they're using those drugs. It's often not possible to tell people, oh, you've got to throw this stuff away because they're addicted. But you can provide messaging that makes them safer. So for example, if they're taking a drug that might have a high level of a toxic component like fentanyl in it, they can take a smaller amount of it, or they can take it slower. Okay. It can also help harm reduction people to know how to treat people. So for example, right now there's a big problem in the availability of a drug called xylazine, which is being found in injectable opioids. And xylazine is a sedative that's used in veterinary medicine for large animal sedation. When it shows up in our in the drug supply, it has side effects in humans. So for example, it can cause really horrendous damage to tissue. So people get these awful wounds. And so if harm reduction people know that xylazine is in their local drug supply, then they can hand out wound care kits. Then maybe people won't have to have amputations. They can treat the wounds before they get to the amputation stage. It's also really important for public health surveillance and response to know what kinds of substances are in the illicit drug supply. And as I will show in a minute, it can impact the kind of treatment that is needed. If somebody's trying to get off of illicit drugs, then you need different treatments if they're addicted to an opioid versus to, say, a benzodiazepine. Okay, the same treatment methods don't work. We work with illicit drugs. We have a modification of the pad that's optimized for detecting illicit substances. So we call that the ID pad. And here's examples. Here's two examples. In one of them in the center has been run with one drug combination. You can see that there's an ID pad here, and then right next to it there is another little microfluidic device. That is a lateral flow immunoassay test strip. So it's kind of like the COVID tests that we all came to know and love. It is a little different from a regular COVID test. So unlike a COVID test, these drug tests for illicit drugs are testing for very small molecules. So they're all run as competitive immunoassays. And so what that means is that if you have one line there, then that's just showing a control line. That means that there's fentanyl present. And in this other example here, where there's two lines, the presence of the test line means that there is no fentanyl present. Okay, so it's kind of the opposite of what we're used to from COVID tests. But that by combining what we see from the colors on the test card and the results from the fentanyl test strip, it's possible to learn about the bulk drug that's present in a sample, but also the fentanyl, which can be present at very low concentrations, can also be detected. So again, we have a phone app that we use to read the ID pad. And then, once we get this information, we still have more questions. So if we know that there's heroin and that it's positive on the fentanyl test strip, we don't know at this point if it's a plain fentanyl or a fentanyl analog. So CDC has a kit of over 251 synthetic opioids, of which 213 are synthetic fentanyl analogs. And of those fentanyl analogs, a test strip will detect between 50 and 80 fentanyl analogs. And so just because you have positive doesn't mean it's fentanyl. It might be one of those designer fentanyls. We don't know how much is present. And we don't know if there's other substances that are present, like xylazine or levamisole, that might also have medical impacts downstream. So all of these questions can be answered if we can get a sample for mass spec. And what we found is that if you take a test strip, so here's a test strip that I'm showing on my camera here. If you pull the, there's like a little sticker. There's like a little sticker that's covering up the active sample absorbing part. There's the little pads in there that are made of absorbent cellulose. So we can just take those out and soak them in acetonitrile or methanol and extract samples for mass spec. So we do the mass spec analysis using tandem mass spec. So I like to tell my students that that instrument is about the cost of a house. It's definitely not a field portable instrument, but it is so much more powerful, right, than any field portable device. It works in complicated mixtures, it tolerates strange cutting agents, and it has really impressive ability to quantify targets down to below nanogram per mil levels. And I know many of you here have experience with tandem mass spec, so I don't need to sing the praises too much, but we've been really hopeful with that. So tandem mass spec, it works because you have basically 3 mass specs in a row. The first one, you have a sample that comes in, maybe in this case, our target is cocaine. The cocaine comes in, it could come in just directly injected, or it could be injected from a mass, an LC or a GC column. And it has to get ionized in a lot of systems. That's done by protonating it. Most of the opioids are tertiary amines, so it's really easy to protonate them. The mass of the protonated cocaine is 304 AMU. And so the first quadrupole can select that mass ion and pass it on to the collision chamber. So all the other drugs that have different weights will be rejected at that point. We're only going to have cocaine in the collision chamber. And then in the collision chamber, we can induce fragmentation, and the fragmentation forms specific pieces, right? And then we can analyze those pieces in the third mass filter. So this is called the multiple reaction monitoring method, or MRM method. And it's very sensitive, very specific analysis method. And so with that, we can generate a calibration curve. You can see at high concentrations of, in this case here, we're calibrating on fentanyl. You can see that this calibration curve is quite linear at low concentrations, and then we start to saturate the detector. So, but this allows us to quantify as well as detect qualitatively. Sometimes we can get absolute concentrations, sometimes we can get ratios. And so that gives us answers. So let me show you some data that we got. This is from samples that were collected in Chicago, mostly in 2021, so right after the pandemic. And this was at a time when we didn't know it, but there was a big change going on in the composition of street drugs. So we analyzed for 22 different high potency illicit substances in these samples. And these are the most common ones that we found. In almost every sample, we found diphenhydramine. In almost every sample, we found fentanyl. And this was a time when the amount of samples with heroin in them was starting to decrease. So if you went today to Chicago and bought street drugs, there would be almost no heroin in those samples. So we kind of caught that on the kind of partway through that process. The users who were using these drugs said, most of them said, more than 80% of the people who supplied these said that they suspected that there were benzodiazepines in the drugs. We only tested at the time for alprazolam, and we didn't find those. We didn't find it. So alprazolam is the active ingredient in Xanax. When we went back in 2023, after developing a method for benzos, we found that more than 80% of these samples actually did contain designer benzos, mostly alprazepam and other designer drugs that are not approved for sale as in the pharmaceutical supply. And this is really important because when people are trying to quit a drug, there's often ways to treat them medically. So for opioid addiction, there's drugs like buprenorphine or methadone, but those drugs don't work on benzos. So to treat people who are addicted to a combination of opioids and benzos, you have much better results if you know that they're addicted to benzos and can treat the benzo as well as the opioid. Okay, so we didn't figure this out until 2023. So, okay. So I think at this point, I want to stop. I want to just put up the picture of all my wonderful students and see if maybe we can have some discussion, and I will stop sharing, if that's okay. Okay.
Well, you let me ask my question that I'm concerned about. So anybody else have some questions?
I have a comment. You probably are aware of this thing called paper mass spectrometry.
Yes.
That's from the Purdue people, I think. They
Yes.
cut a piece of paper into a triangular shape, apply a voltage to it, and put it into an electrospray ionization source.
Yep.
They can generate ions from whatever stuff is on the paper. And your
Yep.
sample is ideally suited for this. I'm gonna
Yep.
if you have tried it or you
I don't have a paper spray ionization source, but I was recently visiting the University of University of Victoria, which is in British Columbia in Canada, and they have a big drug checking operation there. And so there's a storefront downtown where people will bring in samples of street drugs basically for chemical analysis. And the method that they use is a paper spray ionization that feeds into a tandem mass spec. So Michael, your idea is actually being used for drug checking. Yeah. And yes, I would love to try the paper spray ionization, but I just I don't have the mass spec right now.
might
So
have to sell your house.
I would have to sell my house. Okay, question from Anne. Why did they put Benadryl in heroin? So good noticing that diphenhydramine is the active ingredient in
Yes, yeah.
Benadryl. I I I am not sure. It's not uniform. So in West Coast, it's more likely to be mannitol. So, but I think the diphenhydramine is kind of a stimulant. And so it may partially, so often when people are taking opioids, they'll also take stimulant drugs like things like meth or cocaine. That's called speedballing. And the stimulant is to counteract some of the unwanted effects of the opioid. And so
Yeah, I thought that the diphenhydramine was the opposite of a stimulant.
was
Made you
it,
sleepy.
so
I've used it when I
it
had a
knocks you
cold
out.
and I took some to help me sleep, you know.
So
Help
maybe
stop
it's
coughing and sleep better.
maybe it is in there as so that's a good, that's right, yes. So maybe it would be in there as as a depressant, right? To sort of to be
To relax
synergistic
you, yeah.
with the opioid.
Mhm.
Yeah. Yeah,
I
it's
have a question about, I went to your website for the paper products you could, test strips you can get. And
Yeah.
what I'm curious about is using them for like elementary school demos. I do a bunch of those.
Yeah.
And this would be such a great activity to do. So do you have any of those kinds of activities, kind of planned out, or, which strips might be good? And obviously I don't want to bring heroin into the 6th grade to, like you were saying, Marya said that she
Yeah.
teaches a forensic lab where she actually does work with controlled substances. So, that wouldn't be so good for the K6, but
yeah, no.
have you done any of that, or do you have any of that?
Yeah, we do. I do bring those pads to like I mostly have done like high school demos. And there we will test you can test substances that are like over-the-counter substances or kitchen chemicals. So like starch, cornstarch works great. You can do vitamin C tablet, for
Mhm.
example. The ascorbic acid will show up in several of the lanes. So yeah, why don't you, I'm gonna put my email
Great,
in
thank you.
chat and just email me and we can talk.
My daughter, who
Yeah.
is taking CSI, and one of her classes this
Mhm.
semester is fingerprint, analysis. And so she's come to a bunch of these demos with me. So she wants to kind of develop a, little fingerprinting activity for some of the grades. So,
Yeah.
that'd be fun to work that into it and have a little forensics unit.
Yeah, no, that is a really great activity to do. Just even, getting the students to like do an ink print and then they can look and see the minutia.
Exactly. Find all of the, the
Yeah.
ridge analysis is the
Yeah.
friction ridge analysis is the title of the course. So yeah, all the loops and the whirls and the
Yeah. When 2020 hit and we all got sent to our rooms, I worked with some folks to develop lab activities that students could do sort of in a kit form. So we actually have like a paper titrator. So that might be a good, so one way you can do that is you can load the titrator up with like red cabbage juice and then put different household substances on the different little squares. And so that's kind of a low low sample volume way to do that kind of experiment. Did I patent this technology? We have a patent for the paper analytical device. It's what I would classify as a defensive patent. We didn't want somebody else to patent it and then like we would be shut out of using our own technology.
Yeah.
So I can tell you that so far it has not resulted in any I don't have a fancy car in my driveway because yeah.
I have a very brief question, and that has to do more with, is there a chance that you could modify the assay to do sort of a 96 well plate kind of spot assay? That, because I know that there's a program called SpotCell that we can use for reading 96 well plates, and look at essentially color plates. So it's perfectly usable when we're doing, for example, testing for the presence or absence of somebody with SARS. There are many laboratory techniques that can be used, and I'm wondering if your process can be modified for 96 well plates. That's one question that I have. The other question that I have is also, I'm particularly interested in microfluidics because I teach high school students. And one of the things that I want my high school students to do is to develop a bunch of very low cost microfluidic assays that can be utilized by students in countries that do not have the resources that we have in the United States. And this is one of the things that I'm particularly interested in developing chemistry lab-based analyses that can be carried through microfluidics. And I ran into a graduate student at the National Science Teachers Association meeting last, I think it was October. And this was something that fascinated me. She was trying to develop acid-base assays and titrations using microfluidics. So
yeah, that was
anyway,
that was Rachel Roller. She's
was it Rachel? All right. Okay,
yeah.
So I have met her, and so I'm in that headspace right now of trying to see if we can expand that idea for my students to get into the into thinking about developing these very low cost assays for third world countries, where education is limited by resources.
Absolutely. Yeah. And we do, so for those titrators, you can do redox titrations on them. So we have a titration for vitamin C tablets. So you can see if it's got the right amount of vitamin C, but you could also do that for like bleach potency to make sure again, during 2020, bleach was very the thing. So yeah. I think
ironically, I just wrote a vitamin C redox titration for middle schoolers and sent it. It's with AACT right now. So
yeah, super.
But I really want to have that microfluidics part developed for third world countries. So
yeah, I would love to, just again, it's like send me an email, Carolyn, and I will. Because I can stick a bunch of these titrators in an envelope and mail them to you. And if you think they're useful, then we have a website, you can actually go and buy them.
Yeah, much appreciated. Thank you.
I'm wondering, Michael, are you about ready to issue the questions?
Oh, Michael, I hope that wasn't one of the questions that you put in the chat.
Yeah, that's all right. We have more. We have more. Okay, so I'm gonna start
the contest. Michael,
that was the contest. I'm not eligible.
No. You're not eligible, but it turns out that even though all the speakers are not eligible for the contest, they do get the prize at the end of the year when I get my act together.
Okay, the first one is on this way.
Now remember, send the answer to Michael. Not to Maria, not to me, not to everybody, just to Michael.
And a minute later, I'll send a number two question.
Maria, this has been so fun.
Yeah, it
So
was
fun.
for me too. Yeah. Good.
Okay, well, that's the reason I went into chemistry in the first place. I thought it was fun.
I've really enjoyed the developing this test card has made me go and read a lot of the older literature because those chemical color tests were much more common before 1950s, right? So the first IR spectrophotometer that was commercially available was, I think, 1953-ish. And that was followed by NMR spectrophotometers. And at that point, all the organic chemists in the world were like, let's go do this. And
Yeah.
but in that older literature, there's still some really amazing reactions. So,
Speaking of older literature, I remember that in the old literature, not as old as what you're talking about, but I remember that George Whitesides went through a sort of microfluidics phase,
Yep.
and I'm wondering, were you at MIT at the time he did that,
No.
or were you not?
No. That was well, I don't know when he started doing microfluidics, but the paper microfluidics, that work came out in 2009 was the first.
But he was doing applying microfluidics to other settings.
Yeah.
Previous to that.
Yeah. I mean, I've always been so impressed by his versatility and just the range of his sort of intellectual range. But yeah, no, I think that was sort of the first time I saw something that was sort of directly relevant to something I was trying to do.
Yeah. Michael, did you send the second one?
I sent all three.
Oh, okay. All right. Everybody, you have your chance now. And then Michael does the grading, and then we have another volunteer who she is responsible for sending out the prizes. And so, it's a chance, no matter where you live. If you live in Boston, you can still win. It's okay. Before Mina came to the United States, she was in Canada, and I think she won. So we send the prizes all over. If there aren't any further questions, then I guess
I have a question. How do you deal with inorganic fillers or fakes, whatever?
Oh, like things like talc or calcium carbonate?
Yeah, chalk. Yeah.
Yeah, so the chalk shows up on the pad because we have an iron lane. So you get iron carbonates and hydroxycarbonates. So in some cases we can actually see those things. Bye, Mirinda. Yeah.
Can I ask a question? So thank you very much. It was really interesting. So I wonder about the image analysis, because when you get this result, you have the stuff separated by color, then you use an app to take the photo of it. And so there is a computer analysis going on, and based on that, like it gives you a % of what's in there. So I'm wondering, can you comment on the image analysis?
So the image analysis is done, we try to do the image analysis in two different ways. So one is we use a neural net analysis, and the other is we are doing what I would think of as region of interest analysis. So that's the principal component. So for the principal component analysis, we're actually segmenting the image. So each lane is cut into like 10 pieces, and then we measure the RGB color intensities in those different segments. So the bottom segment is what we call the swipe line, which is where you put the drug. So like for the ciprofloxacin, we expect a particular blue color in lane D. And so if we get that color, then it's going to score high on the ciprofloxacin principal component. Right, and then if it also has the orange color from the 1,3-dicarbonyl in lane L, then that'll put it even higher, depending on the intensity of those colors. Then that is related to the quantification.
Right. So basically, it's like a library of known material. Like we know this specific compound gives orange-red color, the other one gives orange blue color or whatever. So that's kind of in the memory of we have a reference, and then we compare what we get with the reference, and based on that intensity, we say this is this, that's that. But what if we have compounds that generate the same color? Then what happens?
Then you can get a false response. So for example,
Okay.
there are other fluoroquinolone antibiotics, so things like ofloxacin, where the colors look very similar to my eye. So we haven't done but there's there's some cases where, so for example, ampicillin and amoxicillin differ only by one OH group. Okay, but their color barcodes are completely different, and they're very clearly distinguished both by eye and also by the image analysis codes. So it depends on the chemistry, and it depends also on us having the samples available so we can train the neural net. So one thing I was surprised by is that when we run lactose and we run microcrystalline cellulose as null samples, they don't have any functional groups that react on the pad, but the neural net can tell them apart. I have no idea what the neural net is seeing, but it's more than 90% accurate.
Wow.
Distinguishing those what I think are unreactive excipients, but maybe something about the texture of the cell. I don't know. I don't know what it's saying. It's a neural net, So,
Very interesting. Thank you very much.
yes, thanks, Mina. Good.
Any more comments? Oops. Well, if not, I'm going to close the meeting, and I invite you to come to the next one, which will be in September, and it's going to be on sustainability. Okay, I'm pretty sure it's going to be on sustainability, but I haven't worked out the details yet. Okay. Thank you all for coming and participating.